That the lifestyle of some male homosexuals has triggered an epidemic of a rare form of cancer.
Infected with HIV is 40% higher than what has been assumed.
1 in 4 teenage girls has a sexually transmitted
News.
disease.
A new report said only a third of people who need life-saving HIV drugs are getting treated worldwide.
In the 1990s, more than 100,000 New Yorkers died from AIDS.
There are an estimated 91,000 people living in the UK with HIV. This includes a figure of 22,000 people who are estimated to be infected but are undiagnosed.
A few months ago, in a biotechnology research centre in Spain, the first major steps were taken towards producing a viable vaccine to eradicate one of the most prominent diseases of the 21st century. So how does it work?
The MVAB HIV vaccine is an altered or attenuated smallpox virus which cannot replicate in our cells. The vaccine particles contain genes inserted from HIV, causing it to display HIV-specific proteins on its surface. An immune response is provoked by these particles. They are engulfed and broken up by dendritic cells, which then present the HIV-specific proteins. Helper T cells with receptors specific to the proteins bind to them. However, finding T cells in the body with receptors that fit these HIV proteins can take a few days. When the helper T cells have bound, they release cytokines, which activate other white blood cells. T white blood cells differentiate into T killer and memory cells. Helper T cells also activate B cells, which form memory B cells and plasma cells, able to produce antibodies specific to the displayed HIV proteins. The plasma and T killer cells which were produced are employed in removing the vaccine from the body and are short-lived. However, memory cells, able to recognize HIV proteins, remained in the body for a year or more in 85% of people tested. This means that in a person infected with an HIV virus displaying the same proteins as the vaccine, the immune response will be much faster, and the individual will be more likely to beat the infection.
HIV infects T white blood cells, incorporating its DNA into theirs. Developing an effective HIV vaccine is extremely difficult because the virus attacks and hides within the very immune cells that would otherwise destroy it. HIV occurs in many forms and is constantly changing, which any treatment needs to be able to keep up with. Although the MVAB vaccine is only effective against the European HIV subtype B, Dr. Esteban tells us that his laboratory have also developed the vaccine MVAC from subtype C prevalent in Asia and Africa. But these treatments are not going to be available anytime soon. The MVAB vaccine is still in small-scale phase 1 trials using only healthy volunteers. Dr. Esteban comments that research towards the vaccine that reduces infection by at least 50% in a much larger group of people, being optimistic, will take between 5 and 10 years. Currently, the vaccine also requires repeated booster immunisations to maintain protection. MVAB is obviously not yet perfect, but it is a step in the right direction.
Testing of the MVAB vaccine was initially undertaken against simian immunodeficiency virus in macaque monkeys and was found to be 100% effective in creating an immune response to SIV infection, although this has been the case in other vaccine trials subsequently found to be ineffective against HIV. Phase 1 testing in humans was then able to proceed. Amongst 30 uninfected volunteers, a very small group of people, 6 were given a placebo, and of the remaining 24 volunteers, 22 showed a strong immune response against HIV. However, immune responses against HIV in a petri dish do not necessarily mean that the same responses would occur in the body, a problem also encountered in previous vaccine trials. Future testing needs to ascertain whether a therapeutic effect is seen in HIV sufferers, reducing viral count and progression to AIDS. In this instance, HIV cannot be removed. The viral genes would remain within a cell's DNA, but would be controlled by the vaccine. Only if MVAB succeeds in these initial therapeutic trials can it go on for more rigorous testing as a preventative HIV vaccine. The effect of immunisation is increased if certain DNA is also injected, but this increases the complexity of vaccine development and use.
The testing was carried out on 30 HIV-free individuals as the vaccine was still part of Phase 1 trialling. The principle behind the vaccine is to train the immune system into remembering the virus. 24 of the HIV-free individuals received the vaccine, 6 received a placebo. The volunteers were inoculated, and over the next few months, blood samples were taken periodically to see the immunological reaction. Researchers found increasing levels of lymphocytes, specifically CD4+ and CD8+, proving that the vaccine was boosting the immune system's response to HIV. As a result, 90% of the treated subjects had a strong immunological response. 85% kept up this response for a year.
Current treatment for HIV consists of therapy with antiretroviral drugs, which disrupt replication of the HIV virus. In highly active antiretroviral therapy, or HAART, combinations of drugs are given, which decreases the chance of the virus developing resistance, but can have very unpleasant side effects and cannot totally remove HIV. The drugs may decrease the amount of virus present, but the possibility of infecting someone else is still a very real danger. Because of this, an effective HIV vaccine is seen by some as the only real way that the AIDS pandemic can ever be stopped. The MVAB vaccine is not the only potential HIV vaccine being tested. Since 1987, over 30 prototype vaccines have been produced. So far, all have been ineffective outside the laboratory, and some have even been considered unethical, giving volunteers a false sense that they are protected from infection. And in the case of HIV, prevention is obviously better than cure.